Okay, so this is our presentation on the eukaryotic cell, or more specifically, the Golgi apparatus.
So the Golgi is located outside of the endoplasmic reticulum, and the Golgi handles the products, which are the proteins of the ER, by modifying, storing, and sending them to other destinations.
So the appearance of the Golgi is it consists of flattened membranous sacs called cisternae, and you can see them right here, and they're actually just right on the outside of the Golgi. And the Golgi has a cis side that is furthest from the ER over here, and then it has a trans side that is closest to the ER, which is over here. And the proteins come in vesicles to the cis side, and then they go through the Golgi and exit on the trans side.
So when we first started learning about the Golgi apparatus, it was a little confusing, but when we found the UPS analogy, it really helped us learn about it a lot easier.
So, a way you can think about it is transportation services and their relationship between cisternae and vesicles. So, step one, the ER, the endoplasmic reticulum made a bunch of mail, which you can think of as protein, and it needs to send it out. And step two, transport to the Golgi. So the mailman, which you can think of as a vesicle, delivers the mail or the proteins to one mailbox that collects all the mail, the proteins, which would be on the cis side of the Golgi. And then step three connects at the cisternae. So the mail or the protein arrives at the cis side of the Golgi apparatus and connects at the cisternae, which you can think of as a loading dock. And there the vesicle coalesces or fuses into the cisternae. And then step four, inside the Golgi, the mail or the protein is now sorted by zip code, which is protein type, once it's inside the Golgi, and is secreted into the bloodstream, sent out to a membrane or to another organelle that needs it.
So inside the Golgi, the protein travels inside outwards, so from cis to trans, and the proteins are further being modified inside. For example, when glycoproteins are formed in the ER, they have their carbohydrates first modified in the ER, and as it travels through the Golgi, it removes some sugar monomers and substitutes others, making a variety of different carbohydrates. So here you can see a photograph of a Timothy grass root cap cell. The bright green shapes are the Golgi bodies containing the cell wall material. So non-cellulose polysaccharides are made in the Golgi, and then they are incorporated into plant cell walls.
So, in short, the ER makes proteins that bud off inside of vesicles that carry them into the Golgi apparatus, which fuse with the cisternae, and then it travels through the Golgi to end up on the trans side and to get shipped off to other organelles that need it or into the bloodstream or wherever it goes.
So why was the protein afraid to travel alone throughout the endoplasmic reticulum? Because it was the rough ER.
Thanks
Thanks
for
for watching.
watching.